Good afternoon. Welcome to ScienCell's webinar series. I'm ScienCell research scientist Daniel Li. In our previous webinars, my colleague Dr. Jennifer Tang talked about qPCR basics and how to optimize primer sensitivity and specificity. And Dr. Jennifer Walser shared some advice we have for primary cell culture. If you'd like to review those webinars, you can go to our website /technicalsupport/webinars, or simply Google ScienCell webinars to watch or download the presentations. Today, we will continue to share several technical tips for successful qPCR. Here is what we're going to talk about today. We'll first discuss some technical tips to ensure qPCR specificity in dye-based qPCR. Then we will talk about internal qPCR controls to include for data reliability. And additional technical tips we think important for a successful qPCR. We'll finish our webinar today with a brief introduction of what melting curve analysis is and what it can tell us. Following this webinar, there will be a question session. Our R&D scientists, Drs. Jennifer Tang, Jennifer Walser, and Bonnie Marsick, will be here to address any questions or concerns you may have. On the lower right corner, there is the slide number. When you ask a question pertaining a certain slide, please include that slide number. During the webinar, if you have any questions, please hold your questions until the end of the presentation in our R&A session. For questions, you have the options of typing your question in the question pane, calling in, or raising your hand. Okay, now that we have all the housekeeping items taken care of, let's get started. First off, how can you ensure qPCR specificity in dye-based qPCR? Dye-based qPCR assays, such as CyberGreen, have many advantages in gene expression analysis. qPCR offers a sensitive, reliable, high throughput, and targeted method and serves as the golden standard in gene expression analysis. Compared to other methods, such as reporter gene assays or cDNA microarray, qPCR assays are more time efficient and convenient in experiment design and operation. Compared to probe-based qPCR assays, dye-based systems are desirable because they're highly adaptable and cost-effective. Throughout the talk, you will see a few light bulb icons like this. The icon indicates important information or tips we'd like to share. Now, what are the caveats when performing dye-based qPCR assays? qPCR specificity is a vital factor to consider when doing dye-based qPCR, because the dyes can bind to any double-strand DNA, regardless of whether it is the desired qPCR product, a non-specific amplification, or a primer dimer. Furthermore, all qPCR data analysis is based on a prerequisite that the qPCR efficiency is around 100%. Therefore, before performing dye-based qPCR assays, primer specificity should be verified and primer efficiency must be evaluated. Since primer specificity and efficiency is decisive in whether or not your qPCR assays can be reliable, good primer design could be considered as the most important aspect in successful qPCR. Here, I listed a few basic primer design criteria. Like these primers I selected, good primers should have a GC content of 45 to 60%, contain 2 to 3 G or Cs in the last 5 nucleotides at the 3' end, and end with 1 to 2 GCs to serve as GC clamps. Now let's take a look at what caveats you should avoid when designing primers by looking at this bad example. This primer is not ideal for several reasons. First, it has a low GC content, which may affect efficient primer binding. It also has no GC clamp at the 3' end. Finally, it has a continuous 5 Gs in its sequence, which should be avoided in primer design. Good primers should give a PCR amplification efficiency between 90 to 110%. Furthermore, when doing qPCR assays, because many genes are analyzed at the same time under the same PCR conditions, all primers in the array should have similar melt temperatures, known as Tm. Remember, good primer design isn't easy. Other than the basic primer features I just mentioned, we have a few more tips for you to consider when designing primers to ensure qPCR specificity. These include primer complementarity, primer length and Tm, amplicon length, and amplicon location. In the next few slides, we will take a look into them in details. When designing primers, keep primer complementarity in mind, because low specificity is mainly caused by nonspecific binding of primers, which means binding to similar sequences at random locations on the DNA template. We recommend using the amplicon sequence to run a blast search against the whole genome or mRNA database and see if there's any chance of unspecific binding caused by gene duplication events. In addition, always run an agarose gel to check qPCR products when verifying primer specificity. Other traps to avoid include primer heterodimer, self-dimer, and hairpin formation, as they may significantly decrease qPCR efficiency. In our previous webinar, Dr. Jennifer Tong talked about how to assess the probability of these structure formation. If you'd like more details regarding this, please go to our website to review that webinar. When working with complex qPCR templates such as cDNA libraries, in our hands, an effective way to improve specificity is to design primers about 22 to 26 nucleotides long, with a high Tm around 65 degrees Celsius. Longer primers with higher Tm can reduce primer unspecific binding because the chance of finding similar sequences on the template is lowered exponentially. This is especially useful when using genomic DNA or cDNA as template. When doing gene expression analysis, please keep in mind that many genes have multiple transcript variants. If your plan is to compare expression profiles among samples containing different variants, or if you're not sure which variant is present in your samples, be sure to pick amplicons in a common region shared by all variants. On the other hand, if you know exactly which variant you want to analyze and need to exclude others, you should design primers to amplify a specific region only present in that target variant. For example, we had a specific request to analyze the soluble version of IL6R only. The difference between soluble variant 2 and membrane-bound variant 1 is that variant 2 lacks a small exon 9. So, we designed a forward primer right on the exon 8, exon 10 junction, and it won't give any amplification signal if exon 9 is there. In addition, when working with cDNA libraries, one important factor to consider is to avoid primer located in repetitive sequences, as the multiple binding sites may affect qPCR efficiency. Limiting the amplicon size to 80 to 230 base pairs can be employed for better specificity. If the amplicon size is too long, the PCR will require more time for elongation, which may increase the chance of nonspecific amplification. If too short, there will be no easy way other than sequencing your PCR product to tell if you get is real or merely a primer dimer. Lastly, Avoid GC-rich and AT-rich regions, as they are known to form secondary structures such as stem loops and may lower the amplification efficiency. As you can see from the past few slides, good primer design is complicated and it is quite time-consuming. For your information, ScienCell's GeneQuery offers carefully designed primers for dye-based qPCR assays. In addition, all our primers are wet lab validated by both melting curve analysis and gel electrophoresis to ensure PCR specificity and efficiency. You can get these validated primers individually or in kits to save you precious research time and effort. Like all other assays, to ensure data reliability, multiple controls must be included in each qPCR array. Now let's take a look at what types of controls are required. Most likely you're doing qPCR arrays to compare gene expression profiles among samples. To be able to do that, you will need 4 types of qPCR controls. And they are, for data normalization, housekeeping genes. For genomic DNA contamination detection, gDNA control. For cDNA contamination detection, no template control. And for PCR inhibitor detection, positive PCR control. In the next few slides, we'll discuss each of these aspects in details. When performing gene expression analysis, make sure you have all the controls required for trustworthy and meaningful data. For example, this is the layout of our GeneQuery human astrocyte cell biology qPCR array kit. Like all other qPCR array kits that we offer, in addition to the carefully selected genes regarding different aspects in cell biology, we include 5 housekeeping genes for data normalization purpose, as shown in red, a genomic DNA control for gDNA contamination detection, as shown in blue, a positive PCR control for PCR inhibitor detection, as shown in green, and the no template control for cDNA contamination detection, as shown in brown, all of which are necessary and sufficient to ensure your data quality. To compare gene expression profiles among samples, you need to compensate data variations caused by template concentration differences in these samples, or simply put, do a data normalization. Housekeeping genes, also known as reference genes, are often chosen as references for normalization purpose. There are many of them, and here I only listed 12 most widely accepted housekeeping genes. Please keep in mind that the expression level of housekeeping genes can change based on the cell types and treatment conditions. When choosing housekeeping genes, you should choose the ones you know to bear stable expression level in your study. If you're not sure, we recommend that you use average of multiple housekeeping genes and exclude the ones that show obvious deviation from others. Again, in our pre-designed GeneQuery qPCR array kits, to ensure accurate data normalization, we conveniently include 5 housekeeping genes: beta-actin, GAPDH, LDHA, NONO, and PPIH in each plate. Due to the high sensitivity and indiscrimination of the qPCR method, contamination will also be amplified exponentially and interfere with accuracy and precision, especially for genes with a low copy number. There are 2 main sources of contamination: internal contamination from genomic DNA and external contamination of cDNA introduced during sample handling. A common issue in qPCR gene expression analysis is the gDNA contamination. gDNA may offer extra copy number, thus skew the gene expression level data. To monitor gDNA contamination, be sure to include a genomic DNA control, GDC, in your qPCR arrays. GDC is a primer set targeting a non-transcribed region on the genome. We'd like you to know that ScienCell offers a GeneQuery cDNA evaluation kit, which contains a housekeeping gene primer set to check if the reverse transcription is successful, and a GDC control for you to conveniently check if there is gDNA presence in your cDNA samples for less than $1 per sample. It is very important that you check your cDNA quality before your real qPCR assays to save you time, effort, and money. Like internal genomic DNA contamination, external cDNA contamination also provides extra copies of template. The source of the contamination may originate from gloves worn when opening a cDNA tube, pipettes used to transfer previous PCR products, or even DNA containing aerosols. To avoid this external contamination, good lab practices must be employed. Always wear new gloves and change them often. Set up a room or a bench designated as DNA-free to handle PCR reagents, and only use aerosol-resistant filter pipette tips. To monitor cDNA contamination in your qPCR array, include a no-template control (NTC), which contains a primer set that can amplify one housekeeping gene. When doing qPCR for the NTC, add all of the qPCR reagents except the template. A negative reading from this wall indicates the array is free of cDNA contamination. Even if the primers have been meticulously optimized, some external factors may still affect the qPCR assays. One such external factor is the presence of PCR inhibitors. PCR inhibitors are any substances that prevent efficient DNA amplification. Examining the exponential phase of the amplification curve on the log scale can help you determine if your PCR is inhibited. Fluorescence doubles with each additional cycle in the exponential phase of an efficient qPCR reaction, as curves A, B, C, and E shown here. They are all efficient amplification with various template copy numbers and no inhibition effect. In contrast, a lower slope in log exponential phase, as indicated in curve D, which has the same template copy number as B, would suggest that fluorescence is not doubling and that PCR inhibitors may be present in your reaction. The sources of PCR inhibitors can be certain proteins, such as hemoglobin, some carbohydrates, and some reagents commonly used in sample preparation, such as EDTA, phenol, and ethanol. We recommend including a positive PCR control, PPC, in your qPCR array to test whether samples contain inhibitors. The PPC should consist of an artificial DNA template with preset template copy number and a primer set that can amplify it. Because the copy number is certain, the number of cycles required to detect amplification is fixed. More cycles needed than expected would indicate PCR inhibition. ScienCell also offers a GeneQuery cDNA evaluation kit deluxe version, which includes 2 housekeeping genes, a gDNA contamination control GDC, and a PPC control for you to check whether or not there is PCR inhibitor presence in the system. Again, we highly recommend checking if your system has gDNA contamination or PCR inhibitors prior to your important gene expression analysis to save you time, effort, and money. We have a few more technical tips to share for successful qPCR. When it comes to template concentration, a common misconception for beginners is that the more template there is, the better. On the contrary, too much template can be problematic. For one thing, many qPCR instruments do baseline adjustment using data collected between cycles 3 and 15. Adding too much template and making the fluorescence detected within 15 cycles may interfere with instrument's default baseline calculation. For another, TE buffer is often used to dissolve and store the cDNA template because it offers a suitable pH for DNA solubility. And the buffer component, EDTA, can chelate ions to suppress unwanted nucleus activity. However, this EDTA in TE buffer is a PCR inhibitor. At low concentrations, the inhibition effect can be neglected, but not when you have too much in the reactions. We'd also like to remind you that DNA sticks to plastic, so please use low retention pipette tips and tubes. Last but not least, use master mix of reagents whenever possible to reduce pipetting errors. For example, commercial cybergreen master mix contains cybergreen dye, DNA polymerase, buffer, DNTP, and water. And you can further mix this cybergreen master mix with your cDNA template, primers, and water when it's feasible to do so. This will make your data more consistent and reliable. Lastly, I'd like to talk about melting curve analysis, which serves as a common accessory to qPCR assays. In melting curve analysis, or MCA, the number of peaks is commonly used to determine the purity of qPCR products. In MCA, temperature is gradually increased to melt or dissociate double-stranded DNA products. As the double-strand DNA dissociates, it loses fluorescence. And this loss in fluorescence with respect to temperature is what a melting curve peak represents. The theory behind using a melting curve to determine product specificity relies on the idea that different qPCR products melt at different temperatures to produce distinct melt peaks. Or simply put, each peak in the melting curve represents one PCR product. For example, in scenario A, single product as confirmed by this agar gel would give a single peak. And in scenario B, two products as revealed by this agar gel would produce two peaks. But is this always reliable? And the answer is no. This principle is based on a faulty assumption that the qPCR product is relatively uniform in GC and AT composition. While it holds for most situations, false negative can happen when the AT and GC bonds are not uniformly distributed, as we show in scenario C, in which this qPCR product contains a GC-rich region and an AT-rich region. Although the qPCR product is pure as the gel shows, it generates two peaks in MCA. This is because at 79 degrees, the AT-rich region will melt first, giving a partial loss in fluorescence and producing a peak. An additional peak would be produced for the same product at 86 degrees once the melting temperature of the GC-rich region is reached, resulting in a bimodal melting curve for a single product. And false positive, as in scenario D, where two qPCR products only give a single sharp peak in MCA, can occur when the GC content of one PCR product compensates for its size difference to another product in the same reaction. So they melt at the same temperature, 89 degrees. Although this may seem improbable, we have observed this firsthand here at ScienCell. This is why We validate all our GeneQuery primers by both melting curve analysis and gel electrophoresis to ensure the purest qPCR products. In conclusion, today we have showed you that good primer design is the key to successful qPCR analysis. To save you time so that you can focus on more important aspects of your research, we recommend our GeneQuery validated primers for your gene expression analysis needs. And when doing qPCR arrays, you should closely monitor for possible contamination by including multiple controls. And good lab practices must be employed for qPCR accuracy. And lastly, keep in mind that melting curve analysis alone is not sufficient for assessing qPCR specificity. When verifying primers, you must also check qPCR products by gel electrophoresis. Again, please trust your qPCR array needs for gene expression analysis to GeneQuery. ScienCell offers many pre-designed qPCR array kits regarding primary cell biology, such as neuron, Schwann cell, microglia, and endothelial cell biology, biological pathways, such as angiogenesis, apoptosis, cell cycling, and Notch signaling, and disease-focused kits, such as ALS, thyroid cancer, and renal cell carcinoma, just to name a few. In addition, we can also make custom qPCR array kits to better fit your needs. All you have to do is to send us a gene list. And again, all our primers are available individually. Thank you for listening to our webinar. Now I'm going to take a quick pause while my colleague, Dr. Jennifer Tang, will start answering your questions. After the session, when you close the program, a short survey will pop up. Please finish this survey to get a 10% off on your next GeneQuery order. Again, starting tomorrow, this webinar can be reviewed and downloaded, and the link will be sent to you through email. Now I will hand the microphone to Dr. Tang.
Hi all, my name is Jennifer Tang, and I'll be answering some questions that we got throughout the talk while Daniel gives his voice a break. One question that we have is, can you change the housekeeping genes that are included in the kits? And yes, you can. The kits are fully customizable, and you can change any of the genes that are included in the kits, actually, or you could build your own from scratch. There's a separate tab for that on the GeneQuery main page. And for a limited time, there's actually no extra cost to customize any of our kits either. Okay, another question that we have is, do I have to run an agarose gel after running your kits? And the answer is no. All of our primers have been tested in-house here with more than 10 different types of primary cell cDNA, and we've tested all of our primers by melting curve analysis and amplification curve analysis and gel electrophoresis. So all of that is done for you. You don't have to test them again. Okay, how can qPCR efficiency be over 100%? It's a good question. So when you're calculating qPCR efficiency using a serial dilution curve, if you have PCR inhibitors, then your standards are off, basically, and that's how you end up with over 100% efficiency. It's obviously false. It skews your data. So that's why it's important to run all those controls to make sure that PCR inhibitors aren't in your samples. Okay. So if you're only interested in a few genes rather than a whole kit, can you purchase individual primer sets? And the answer is yes. All of our primer sets can be purchased individually, and those are all on our website. Okay. So we have a question of, do we have a cDNA cleanup kit? And what cDNA concentration do you recommend for your preset kits? We do not offer a cDNA cleanup kit. We only offer the controls to detect whether or not it needs to be cleaned. As far as concentrations per reaction, we recommend 1 nanogram per reaction to start your optimization. And I believe all of our kits are they are optimized to do 1 nanogram per reaction. Okay. So are there other options other than Cyber? GeneQuery is compatible with any dye-based qPCRs such as Cyber and Evergreen. And like other options for qPCR in general are probe-based qPCRs, but we don't currently offer that type because they're less cost-effective. Okay. If there are any more questions, we'll wait a couple of extra minutes to make sure there aren't any struggling questions. But I mean, if you do have questions, even after we close the webinar, you can always send them to our tech support and they'll get routed to the relevant scientists who will answer them for you then as well. Okay, thanks for attending, and we'll see you at our next webinar.